A new plague, mass casualties, social upheaval.
It's mutated.
Which way, better or worse?
Remember when that was just the premise for Hollywood scare films? Our recent brush with a real-life pandemic is the latest chapter in an ancient struggle, an eons-long evolutionary arms race between humans and viruses. And we've done okay. In the long run, our immune systems have evolved to overcome all kinds of flus, plagues, poxes, but we've paid a huge price in human lives. Natural selection is brilliant, but it's slow. Now, for the first time in human history, we have the tools to put it into overdrive. I'm Gabriel Spitzer, and this is Pocket Science from KUOW. Each episode, we comb the Pacific Northwest to uncover the fascinating, weird, groundbreaking science happening in our backyard and hear from the human beings who are figuring it out. Today, we're going to meet a scientist who's working to turbocharge evolution so we can teach our bodies how to defend against the next new virus.
So this is kind of like the big fly ranch.
You can often find Harmit Malik down on the ranch, the fruit fly ranch, in his lab at the Fred Hutch Cancer Center in Seattle. He proudly shows off rack after rack of vials, teeming with these freaky little insects crawling around inside.
It's fun to look at some of these flies because they have interesting mutations like eyes growing on hind limbs and fluorescently growing eyes. They're kind of cool. We use them because these are developmental mutations that we study.
This would give my wife hives if she saw this. Oh my
If you
God.
look under a microscope, it definitely is the stuff of nightmares.
Fruit flies are the perfect little models to study genetics and evolution, which are really what's at the heart of what the Malik Lab is trying to understand. Is that a Darwin bobblehead? No.
That is a Darwin bobblehead.
I'm bobbling him. He looks glorious. But the natural selection that Darwin described takes its own sweet time to create, say, a new species.
For example, when we're talking about genes between two different fruit fly species, we're talking about millions of years.
Viruses, on the other hand, evolve at lightning speed.
When we're talking about genes in viruses that we study, we're often talking in a matter of hours and days.
Viruses are constantly mutating to sneak past our defenses, and our immune system has to constantly evolve just to keep up. This is the major theme in Malik's lab, learning from evolutionary arms races.
An evolutionary arms race, the simplest way to think about it is the opposite of symbiosis. In a symbiosis, you have two partners that can reach an equilibrium where they're each contributing to the other's success because that feeds back to their success. An evolutionary arms race is where that equilibrium cannot be achieved, where the fitness of one species has to occur at the expense of fitness of the other species.
So the balance of power goes back and forth. And that's never more apparent than when a new virus comes along.
For example, the smallpox pandemic.
The virus has the upper hand for a while. It might kill off the weakest and most susceptible hosts, leaving only the fittest to survive.
What that did was it basically put the population through a sieve.
The strongest hosts, or the ones with random helpful genetic mutations, they make it through the sieve and reproduce.
So we are all sort of descendants of human beings that survived these epidemics and successfully passed on innate immune genes that basically afforded them and us now protection against something like the smallpox.
We inherit that protection in our DNA. And it makes up one of the two big layers of defense that our bodies have against pathogens. It's called the innate immune system, and it's like an automatic alarm that kicks in when we have an infection. Its job is to basically slow the virus down so that our other big defense, the adaptive immune system, has time to ID the pathogen and gin up a response. That second immune system, the adaptive one, is probably more familiar. Vaccines, antibodies, white blood cells, these are all aimed at recognizing a specific pathogen and going after its particular weaknesses. But Malik is focused on the innate system that we're born with, the first responders. They're a more blunt instrument, but if they don't do their job, the specially trained defenses might never get the chance to deploy.
You could actually lose the battle in the beginning and the middle, so you never even get to the end game. So you could have a really good adaptive immune system that never actually gets to show you what it's got because your innate immunity was not able to defend you.
Both arms of the immune system are crucial. But for medical science, the innate immune system is still kind of an untapped resource.
We have not really exploited the first wave of immunity very effectively, partly because we just didn't know how to do that. And now we are actually learning more and more about how to do that. And given all the advances in synthetic biology and our ability to actually almost anticipate, if you will, what the next pathogen might be, we can actually set up preemptively some countermeasures in place.
Specifically, Malik wants to leverage the capabilities of our innate immune system to give us a different way to fight new viruses. The first step in exploiting innate immunity is to really understand how it's worked over the eons. And he can do that because the viruses that we've confronted in our distant past leave fingerprints in our genes.
We can see evolutionary echoes of that selection in the genes that we still have in our genomes today.
The human genome is the complete genetic recipe for a human being. It's in each one of our cells. And our DNA turns out to be a big-time hoarder. It hangs on to all kinds of artifacts from past evolutionary contests. Malik compares it to a fossil record of our deep evolutionary history.
For most of the viruses that we study, we actually don't have the virus itself. That virus has left a fossilized version of itself in our genome where we can interpret what that virus must have looked like when it was raging in its infectious peak.
Over the millennia, bits of virus genes get incorporated permanently into our DNA. After a while, it really adds up.
And then once you look with that lens, you recognize that more than 8% of the genome is actually made of these dead viruses.
I gotta linger on that for just a second. 8% of the code in our DNA comes from viruses. You're like 1/12 virus. Yuck.
Yes, most of these are actually not doing any benefit or harm in the genome. Most of the time, these sort of just sit and decay, accumulating cobwebs, if you will, metaphorically. Occasionally, though, just like any antique that you might have in your attic, some of these have been repurposed for very, very important functions.
So we all carry around this vestigial junk in our genomes. And Malik is like one of those Antiques Roadshow guys who can look at all these mothballed genetic sequences and figure out which ones have value. In decoding these old sequences, Malik is looking to understand how our ancestors overcame viruses in the distant past. The hope is to use that playbook to evolve defenses against future viruses, not in a matter of millennia, but in weeks or days.
What's really cool is that this sounds like a little bit of an evolutionary history lesson, which it is, but we have been able to take that history lesson and actually convert that into a sort of a pharmaceutical goldmine. We can actually now introduce mutations not in a random way, but in a much more targeted way in the laboratory and basically unleash completely novel antiviral activities, including converting these innate immune proteins that are somewhat effective against viruses such as influenza into much more potent versions against them.
So the idea is that by learning how the evolution operates, you are potentially able to recreate that, but in a very directed way in the lab.
Yeah, because obviously our ancestors survived those viruses. So if we can actually learn the lessons both from the viruses side, but also from the innate immune side, we can actually then deploy those lessons in the present day time.
Malik's lab does that by imitating evolution, creating thousands of synthetic genes that are almost all alike, except for tiny variations.
And we can basically test them en masse against a particular virus to identify which variants are much, much better at basically defeating this virus, and then use those as the sort of parents of the next generation of this similar exercise. If we had to wait for natural selection to impose these, it would take tens to dozens of generations. But by taking advantage of advances in synthetic biology, we can really jumpstart hundreds to thousands of years of evolution in basically a week.
And what might that lead to? Well, potentially it could be a pill that ramps up our innate immune response temporarily. So say the bird flu jumps to humans. The pill gives our cells a power-up so they can hold off the infection long enough for our white blood cells to get their act together.
So this is the room which is more expensive than my house. This is a
There are a few of these rooms in Malik's lab, not much bigger than supply closets. They're outfitted with fancy microscopes, biosafety cabinets, and hyper-technical refrigerators. Inside sit banks of test tubes where evolution is flying by at breakneck speed.
What we do is we take these cells, we introduce these DNA variants that we made in the lab into the cells, and then we infect them with the viruses, and then we look for the survivors. So that all happens kind of in this room.
I was hoping there would be a room like this. That would be like the room where it happens.
And this is a spinning disc microscope. This is the kind of picture that we can take where that's actually one cell, and we can see like all of the inner features, including the traffic highway of the cell, etc.
It looks like a nebula or something.
It does look like a nebula. Actually, we sometimes have high schoolers come in, and it's the same sort of sense of awe you get from looking in space as you get looking at in a cell. I sort of think of the analogies in the same way.
Do you still feel some awe?
I do feel some awe. I'm actually not on the bench as much, so these are the folks who are doing most of the work. I mostly tend to doodle on my board and come up with ideas or ways to distract them from the work that they're doing. But there is a sense of awe. And sometimes, these things take time. It's not like I get an idea and then we get fulfillment within a year. Sometimes it takes 4 years and 5 years. And in the midst of that, it's easy to lose perspective on that arc. And sometimes it's easy for them to lose perspective. And so my job is just to remind them to not lose that sense of why they got excited about science in the first place.
So progress here can be painstaking and take years, but whether that feels slow or really, really fast to you kind of depends on your perspective. Well, specifically, it depends on how old you are. The technology has come a long way since Dr. Malik got his start.
I remember when I was a grad student, I had to stay overnight in the lab making these samples, but now it's all automated.
Yeah, I don't know how much folks appreciate how much of science used to be like counting things.
But whenever I bring that up, I have that okay boomer sort of look. They don't say that because I'm technically their employer, but definitely the look is that of okay.
When you started, did you imagine that you were going to have this capability to just manufacture millions of genetic variants of your choice and see what they do?
I did not. I mean, I look back actually at papers which we wrote and we were very proud of 2 decades ago. And then we realized those papers would take perhaps an afternoon today. And those took us months and years. It's a very deeply depressing thought. But the point is that actually viruses have had these enormous advantages all this time. And it's not that we've leveled the playing field at all, but we are getting closer. That's actually an enormous boon and blessing. And not taking advantage of that is literally reversing the clock by a century, which just seems like that would be a crazy thing to do.
What has the progress that you have made in those insights that your lab and your colleagues have amassed say about the importance of basic science?
The beauty of basic sciences is that I think its potential is limitless. We have many discoveries that were made not with the intention of providing cures in cancer next week, but actually are providing cures in cancer. Immunotherapy, mRNA vaccines, these are all fundamentally rooted in basic sciences. So you just never know what the importance of that discovery could mean in a broader sense. There will be some dead ends. That's just the nature of basic sciences. But you're basically asking incredibly creative people, perhaps at the peak of their creativity, to really put their energies for a few years in the lab to focus on this problem. And amazing things have kind of come out of that.
It could still be years before we have a pill to turbocharge our innate immune system. But hey, that's better than millennia. And if the COVID-19 pandemic taught us anything, it's that the viruses aren't letting up. This is an arms race, and now, for the first time, we might be able to give ourselves a head start. You've been listening to Pocket Science from KUOW in Seattle. I'm Gabriel Spitzer. This podcast was edited by Jim Gates and produced by Shane Mailing. A big thanks to Brendan Sweeney, Marshall Eisen, and Jason Pagano. Music by Artlist and Blue Dot Sessions. If you enjoyed Pocket Science, tell a friend or write us a review. It really helps. And you can support our podcast by visiting kuow.org/pocket. We'll see you in 2 weeks for another episode of Pocket Science.